NAPNAP members online. I'd like to give a shout out specifically to Pat Shabaro. She just had me on her show this afternoon, the Nurse Practitioner Show on Dr. Radio, and we had a great time. So thank you so much for that. We also have our other immunization guru, Mary Beth Koslap-Patraco. I see you out there. She's talking to somebody, but we see her out there, and everybody loves Mary Beth. So hi, and thanks for joining us. We also have our government consultant, Dave Mason, so our policy guru. So thank you so much for joining us. I know a little thing called the election is keeping you a little busy, I think, in Washington, D.C. following that. If you want to, declare a winner by the end of the night, that would be great and really helpful to all of us. So your insider information, but thank you. And also a shout out to our new executive director, Jim Windorf, who has joined us along with all of you other wonderful people. Just wanted to give a couple of announcements before we get started. And I know you're waiting for the big show with Patsy Stinchfield tonight, and we're excited to share with you information about the COVID-19 vaccine. And you can see by our attendance, obviously, this is something that's generated a lot of interest. As experts in pediatrics and advocates for children, the National Association of Pediatric Nurse Practitioners is here to make sure that you are up to date on the latest information to help you impact pediatric health outcomes. So wanted to give you a few announcements and heads up about some things that are coming your way. Most of you have seen our new branding on our website, and if you haven't had a chance to check out our brand new website, I would encourage you to do so. You can go to napnappartners.org or napnappartners.org, both of our websites there. On napnap.org, we have two really great, relatively new resources for you. We have a COVID-19 safety page with collated resources there. And we also have a child health equity resource page to give you information on how NAPNAP is working to impact child health equity. We have launched our first ever podcast called TeamPeds Talks, with the first conversation series being on child health equity. We are offering to NAPNAP members 8 hours of free continuing education with 16 episodes. And we have several of you I see on here who have been guests on the show. And Darwin, you're right there on the top of my list. I can see you, and a lot of other really great members. So if you are feeling a little discouraged and anxious by looking at the news lately, this is news that you can open that will really lift your spirits and give you faith and confidence in your colleagues across the country who are working to do such excellent things. We will be having our next TeamPeds Town Hall as a fireside chat on Tuesday, November 17th, just a couple of weeks from now. And that will be a get to know you with our new executive director, Jim Windorf. That'll be at 7 o'clock Central Time, and so be watching for that. I also wanted to encourage everybody to participate in Nurse Practitioner Week that is starting this week coming up. Remember that if you're on social media and you want to share anything about nurse practitioners, about what you're doing, our new hashtag that we're using is NPs for Kids, NPs for Kids. And I'm asking Michelle to put that in the chat for you so you can see it there. So be sure and tag us at NAPNAP of what you're doing with NPs for Kids. And we would love to showcase what nurse practitioners are doing for kids. The last thing that I have is our March conference is coming up. Registration has opened up this week, and it is a conference your way. After doing an extensive member survey, we are offering an in-person conference and a virtual conference, whatever meets your health needs, your organizational requirements, or whichever way you feel like is the best way for you to learn. You can join us in person in Orlando in March, or a week following that, we will have the entire conference online in a virtual format, and you will have online access on demand until July 31st for that, which is really exciting. So lots going on. If you're not following us on social media, make sure you do that at NAPNAP, at NAPNAP Partners on Facebook, Twitter, and Instagram. And I am going to turn it over to you, to our host for tonight, our immediate past president, Raji Kapolu, who is going to introduce our guest, Patsy Stinchfield. Thank you so much for everyone for attending, and enjoy tonight's town hall.
Thank you so much, Jessica. Everyone can hear me okay? Okay. Well, it's so wonderful to see everybody, and thank you so much. It looks like we have almost 140 people here this evening, so thank you for taking the time to be here. This is our third town hall officially this year. We had the opportunity in the spring to hear from frontline care providers in primary and acute and critical care about their experience with COVID-19. More recently, we hosted a town hall on the impact of racism on children and adolescents, and now this town hall this evening to talk about COVID-19 and specifically the vaccine and its development. And so we really enjoy bringing together our members in the community to talk about these relevant and timely topics as they come up. So just a reminder to everybody that we are recording the webinar so that it can be available in the future. You can feel free to share your video. I love to see everybody's faces, but certainly you may turn it off if you would like. After I introduce our incredible speaker and she has the opportunity to present, I will start a dialogue with some of the pre-submitted questions from all of you before today, and then toward the end of the call, we'll open it up to general Q&A. So if you have any questions or comments, just type them in the chat box as we go along. I'll watch very carefully and we'll do our best to get to as many questions or comments, although I think our speaker will do a wonderful job addressing many of the things that I know we're very curious to learn about. So with that, I personally, truly, this is an absolute honor to introduce our speaker this evening. And I'm just so pleased to welcome a NAPNAP member and nationally known infectious disease and immunology expert, Patricia or Patsy Stinchfield. Patsy is a board certified pediatric nurse practitioner with over 33 years in infectious disease and 42 years in nursing, who specializes in vaccines and vaccine preventable diseases. She is board certified in infection prevention and control and serves as the Senior Director of Infection Prevention and Control at the Children's Immunization Project at Children's Minnesota. Having led the Children's Hospital Incident Command response to the measles outbreak in Minnesota in 2017, Patsy is now helping to lead the Children's Minnesota response to the COVID-19 pandemic through the Hospital Incident Command System as Deputy Incident Commander and Chief of the Medical Technical Branch. Patsy is widely recognized as a specialist in vaccine preventable diseases, having served as the first nurse voting member of the CDC's Advisory Committee on Immunization Practices, ACIP, where she continues as a liaison representing the National Association of Pediatric Nurse Practitioners. She is a longtime member of the CDC's ACIP Influenza Working Group, the Pediatric Schedule Group, and Combination Vaccine Working Group. Patsy is president-elect of the National Foundation for Infectious Diseases, NFID, whose mission is to educate clinicians and the public about infectious diseases and how to prevent them. She is a regular national and international lecturer in pediatric vaccine-related topics and often sought by local, national, and international media for interviews related to infectious disease and immunization. Testifying at the Minnesota State Legislature on vaccine preventable diseases and vaccine exemptions has been a regular role for Patsy over the years. Patsy is happily married and most proud of her 2 fully vaccinated adult daughters and 1 granddaughter. Patsy, it's an absolute honor to welcome you this evening, and I turn the floor to you. Thank you so much.
Great. Thank you so much, Raji. I have anything to disclose. I am a ACIP member, so we will be at one of the last stops for COVID vaccine approvals. And many of the slides that you'll see tonight are from a recent ACIP meeting. I also want to thank my colleague, Dr. Amy Snevice at Children's Minnesota. So this is what we're going to hopefully get through tonight, talk about the epidemiology of COVID, talk about the general understanding of the virus and the immune responses. We'll do some clinical updates, but mostly really focusing on the vaccine. And I have included the questions that you have submitted in advance. But as you know, you can't have a COVID talk without this slide. The epidemiology is not getting any better. The United States hit that long predicted over 100,000 cases per day that Dr. Fauci predicted. We hit that yesterday. So the United States continues to lead and in cases and deaths, and it's really quite worrisome. When you look at it from a pediatric standpoint, this is from the CHA and American Academy of Pediatrics. The blue boxes are the cumulative cases up through October 22nd. And so you can see some of the heaviest hit cases are in that dark blue: California, Arizona, Wisconsin. Sounds like the political map here. And then the percent increase in child cases in the green, and those are the areas, primarily the Midwest, as you can see, that have been increasing recently. And this slide I really like because it points out, this is in Minnesota, and it points out our what we all know about COVID so well, which is our high impact to our people of color. So Minnesota is predominantly a white state, as you can see in the legend there. 80% of our population is white, non-Hispanic, and just 6% is Black, non-Hispanic. But when you come down and look at that population, while they're only 6% of the population, they're 14% of the proportion of cases, 18% of hospitalizations, 17% of our ICUs, and 9% of our deaths. So it's really quite problematic. This is also a very helpful slide. When you look at it, these are ages across the bottom. And so the proportion of cases, we're really seeing these high numbers in these young adults, 20 to 24. And we all know about child and adolescent development. And so you can sort of figure out why that is happening. And then this lowering down as in proportion of cases as you get older. However, the hospitalizations go up as we age, into the peaking here in the 60s to 70s, and then down. The intensive care ages really shifts more to the right, closer to late 60s, 70s, and then goes down. And then the proportion of deaths, as we all well know, is this higher into our 70s, 80s, and 90s, where most of our mortality is. We have COVID-19 hospitalizations and death. I really like this for perspective. If you take that same young adult as your comparison group, and then you look at how do other age groups for hospitalization compare, young children are 4 times lower than those 18 to 29-year-olds, and 5 to 17-year-olds are 9 times lower. Whereas when you look into the older age group, 65 to 75, 5 times higher. Same thing with death. As you get older, those percentages or proportion compared to the comparison group just goes up and up. So I wanted to answer some questions people had about transmission. Pediatric transmission is interesting. Kids do transmit this virus. And what we know is that about 9.8% of COVID cases in the US are in children, and about 2% of them have hospitalizations, whereas less than 0.5% have deaths. And this is all important data to know as you're thinking about vaccines and what population we're going to be focusing. Where are the cases? Where are the deaths? What are our goals with COVID vaccine? So as you can see, this is more recent. This is September. I really liked this MMWR that talked about transmission dynamics of COVID. And this was in looking at Salt Lake City. There were 12 kids in 2 childcare centers, and there were infections that were known to be transmitted from children to adults. So the kids transmitted to parents. There was an 8-month-old that transmitted to both of their parents who had not been ill prior, but the baby got exposed and sick at the daycare. So it was a good way to document from infant to adult. 2 of the 3 kids that were asymptomatic were confirmed as kids that spread it. One of the adults that got it ended up hospitalized. So I think it's an important message to us related to transmission, that it can go both ways. It's more commonly adults to children, but it can go children to adults. And this is a very recent MMWR from October 30th with the same message here. That transmission, this was looking at Tennessee and Wisconsin between April and September. And the findings on this prospective household study, where they really looked at these families intensively, that the household transmission from 101 index patients was common and occurred early after their illness onset. The secondary infection rate within a household is about 53%. So household transmission, we think, is really one of our big aspects of spreading COVID right now. So when you think about the clinical manifestations, this again, this legend here of what is our goal with vaccines? This is a bad disease. It's worse than influenza, many, many more deaths than influenza per year. But what are we trying to accomplish with vaccines? And that's an important sort of target message in the beginning. Do we want to try to eliminate all COVID or just the severe and critical in this 10%, the moderate, mild? What's our goal overall with vaccines? And you can see most cases of COVID are in this asymptomatic, mild to moderate, and as opposed to 10% in pediatrics in that severe and critical. The other thing you can easily see in this is not just a respiratory illness as we first thought in way early last winter and spring. This is a virus that really likes all organ systems and can cause lots of trouble. This is also a big question that came up through the early questions from all of you is what about this whole twindemic, COVID and influenza? How do we diagnose COVID versus influenza? What about vaccines and influenza? And we know that there are just so many features that are similar between COVID and influenza. It is really, really difficult to diagnose it. One of the most important things I think will be just taking a great history. If it's a slow, sort of subtle onset, a few days ago had a runny nose, sore throat, and they kind of didn't feel so good, that's probably less likely to be influenza. That sounds more like COVID, whereas influenza tends to hit fast, hit hard. And so the onset of it, I think, is a really important distinguishing characteristic. But when you look at this clinical characteristics, there were similar rates of hospitalization and admissions to ICU between these COVID and influenza. Less than a year old and greater than 15-year-olds are more commonly hospitalized with COVID. Fever, diarrhea, vomiting, headache, body aches, chest pain were found more often in hospitalized children with COVID. So there are definitely some differences. And then MIS-C, I think for our pediatric providers, this is the thing that probably keeps us up at night, is worrying about these kids that they may have been asymptomatic or a mild infection, but that down the road, usually within that first 90 days or so, that you can have a child develop MIS-C, the multi-system inflammatory syndrome in children. And this is a concern because it can have significant impact to multiple body systems, as you know. The clinical characteristics of this are interesting. It's about 8.3 years of age is the median age. Only about a quarter of them had underlying medical conditions. So it's not just immune suppressed or kids with other chronic illnesses. 80% of them required an ICU admission and mechanical ventilation. The good news is that 70% are fully recovered in this study, and there were 4 deaths. So my institution at Children's Minnesota, we've seen about 30 MIS-C children in the state of Minnesota, and we've seen at Children's where I work about 22 of those 30. So we are getting experience in this. And as you can see, the MIS-C clinical manifestations, primarily heart, you've got myocarditis, cardiomyopathy, you get vasculitis, hypotension, and they really present very sick, very shock-like. GI, 91%, mucocutaneous rash and 72%, and then lots of hematologic problems. So I wanted to just mention that because these, again, are reasons you want to vaccinate. It's why we want to have us think about vaccines for children against COVID at some point in time. The other thing we have to think about clinically is what happens in our immune system when we have the disease, and because we can't expect a vaccine to outperform our own immune system. And so understanding how our immunology fights the disease is very important. So immunity on the disease side here, what is known is that most COVID-19 patients do mount an IgG and IgM response to the virus, and many COVID-19 patients mount neutralizing antibody responses, so a little more lasting And the magnitude of antibody response correlates to the disease severity. So the sicker you are, the higher you're going to have your antibody levels. What we don't know is, are COVID-19 patients susceptible to reinfection? There's a handful of reports in the literature that this can happen and can be worrisome. And are antibodies a correlate of immunity, and how long does that immunity last? We really don't know. Most coronaviruses, the immunity, the antibodies do not last very long. So we're learning a lot still about the disease and immunology because it's still so new. We don't have a long-term experience. Genetically, it's important to understand how the virus works too, and I think this is a few things that we do know, that SARS-CoV-2 has had a very slow genetic drift to date. When you think about influenza and how it likes to be so wily and change itself even from the southern hemisphere to the northern hemisphere, this virus does not seem to have a rapid genetic drift. It seems to be a relatively stable genome and low genetic diversity, making it pretty well suited for a vaccine. There are differences in the glycoproteins, and that's for those who are making vaccines, that's an important part that they're following. And then that mutation rate is just about a third of what we see in influenza. So let's get into the vaccine aspects then. Thinking about, we're going to kind of walk through a little bit cellular, a little bit philosophical, ethical, and a little bit public health interventions here. So one of the questions was the question was kind of, who's in charge here? There's a lot of people involved in vaccine development, and it does seem like there's an alphabet soup of involvement. And so this is the path from clinical development to recommendation. That there is the need to develop a vaccine, and this is the various clinical people, and then we've got our warp speed office that has developed the United States mandate for this, and then the manufacturing of the vaccine. Can we do that? How can we do that fast? That goes then, FDA is our body that does the licensing. Most vaccines are licensed before they're used. In this case, we are considering this emergency use authorization, where we have enough study in to know that it's safe and it's effective, but it may be used before it's licensed. But there are definitely clear safety thresholds that have to be met. Another pathway that FDA has talked about, and VRBPAC had a conversation about this, which is expanded access IND. You think about when Oregon had the big meningitis outbreak, and the MenB vaccine was not quite finished, but was enough developed that and enough study that they felt comfortable releasing it early in an expanded access. And so that's another possible pathway. The ACIP, Advisory Committee on Immunization Practices, which I represent NAPNAP on, is that committee that reviews the evidence, looking at the recommendation framework and making those recommendations back to the CDC director. So we are an advisory group. We don't make final decisions. The final decision then, as you can see, goes to the CDC recommendations for post-approval monitoring. All of these bodies, and then NIH, of course, with NVAC, there's advisory committees on all of these, and I've been very impressed with the coordination. It does seem like all of these entities are working well together. And this is just another way to look at it, that the manufacturers play a significant role in putting out the clinical research, scientific base, going through the FDA. And the FDA VRBPAC committee, I think, is really, really important here. They're a group of scientists, clinicians, some public health individuals, the public, who are really looking and asking a lot about all the safety signals, et cetera. And then again, through the CDC. And then at that bottom part of it, the thing that's going to be really unique with this is the rapid distribution and out to distribution facilities, and then to administration sites. So it's not going to be like your typical vaccine distribution. There will be significant control over that by the state health department. So On the 30th of October, we had our ACIP meeting presentation, and we had a number of presentations. It was an all-day meeting. This was an additional day to our regular October. We meet in June, October, and February. We've had a number of emergency meetings. We just got another one added to our calendar for December. So there's more to think about with COVID vaccine coming up even before the end of the year. The vaccine update summary there was that there's 248 COVID vaccines currently under development. And this is a phenomenal international collaboration, and the number of different countries that are working on vaccines, working together on vaccines, these various candidates across the world. 5 candidates are actively recruiting in the United States. And so those are some of the ones that I'm going to emphasize today. As they said at the ACIP, we have some pre-established expectations. And one of the expectations is that a 50% efficacy is what we want. So that's not very great. We get that with influenza, sometimes higher, sometimes lower. You'd want, really what we want is 80% or higher, but we will take 50%. Versus a placebo with a confidence interval lower bound needing to be no lower than 30%. At least half of the subjects will need to be followed up for at least 2 months, and this got quite a bit of discussion both at VRPAC and ACIP, sorry, that 2 months seems to be a little bit too short as far as the initial follow-up. All the subjects will be followed up for quite some time, but in terms of can we release this to the public and get it distributed, that 2 months, if you're going to see, the people who are fine with that idea are those who say 2 months, if you're going to see a side effect, you will see it within the first 2 months. And others feel like 2 months is not that long before we roll it out to the large population. So that's still, keep your ear to that. That one may have more discussion. And then, of course, safety data in well over 3,000 recipients. So the manufacturers have to show us the safety data, has to be a minimum of 3,000 recipients. We want to look at reactogenicity, what was the local side effects, what were the systemic side effects, were any adverse events, and at least some severe disease prevention. Not just that mild disease, but some severe disease prevention. So here's our list. This was also from the ACIP meeting. And if you look at this, this is the ones that are mostly in phase 3 are what I'm going to emphasize tonight. You've heard about the first one out at the top, the Moderna, which is the mRNA. When you look down on all of these in the trial characteristics, they're almost all 2 doses. They are all in 18 years and older. Some of their upper ages range between mid-50s to mid-80s. Almost all of them are 2 doses. I think the Janssen pharmaceutical is hoping for 1 dose. Novavax also may be hoping for 1 dose. Most of them will be 2 doses. And I think all of them except Janssen is going to need freezing. And so that's one of the challenges. Some of them are like super sub-freezing and in freezers that no hospital or public health department carries. So those of you who have been working on the distribution process in your state can comment on your thoughts on that. So as we look at vaccines, you really have to understand how the basic structure of the virus. And in this case, there's a lot of focus on the spike protein, that single-stranded RNA that is the spike protein. That's where most of the emphasis is. Also this RNA inside this envelope. And so the different ways that vaccines are made will focus on different parts. So the spike protein, the M protein, this inner RNA, and then from those, you look at how that virus enters the cell as this ACE2 receptor. I'm also really kind of following some therapeutics, not vaccines, that are trying to block right here, right between the virus and the ACE2. If you don't allow it to get in systemically, that would be marvelous. Once it gets in, though, it releases its RNA, develops these proteins, and then as viruses do, their whole existence is to make more of themselves. And they have to be in a cell to be able to do that. So I just thought I would pull one slide from one of the manufacturers. This happens to be the Novavax vaccine, just to give you an idea of the kinds of things, the data that we look at ACIP. So phase 1-2 trials are you're determining safety in a small amount of people, and you're trying to figure out the dose. And this is from the New England Journal of Medicine on the spike protein nanoparticle vaccine type. And so just to walk you through this, you're comparing your vaccine candidate against a placebo, and you check them at 0, 21, and 35 days. And then the next thing you're trying to do is figure out the dose. Is it 25 micrograms, 5 micrograms? And so they'll compare 25 micrograms alone, and then 5 micrograms with this matrix, which is an adjuvant. And adjuvants are really important for the success of our vaccines. They are stimulants to our immune system. It helps our immune system recognize that something is present and really triggers a more robust immune response. So adjuvants are very important. 25 micrograms, and then looking at that, this one comparing dose 1 with the adjuvant and the virus, and then dose 2 with the placebo. So you'll take a look here. You can see that there's very little response at the placebo, whereas when you look at both the 5 microgram and the 25 microgram with the adjuvant, that you get this very nice high rise in antibody responses. And so that is what we're looking for. This is IgG antibody. And then when you look at it compared to the right, compared to someone who had COVID and what their own human convalescent serum was, it's impressive that it's even more responsive than if for those who had the disease. So you're looking at those, of course, with asymptomatic, you're not, again, going to mount as much of an antibody response compared to those who are very severely ill in that purple triangle. They are going to have a higher antibody response. One of the things we want to make sure is that these studies are representing diverse communities. The manufacturers are trying to share this on their website. This is just a screenshot from Moderna, looking at how they are being very transparent about who's in their studies. Again, looking at risk groups, so wanting to make sure that we're not just studying this in healthy individuals, but that we're looking at how the vaccine will work in people with high risk complications and comorbidities for severe COVID. I like the fact that they're really paying attention to this, not only race and ethnicity, but even some details that, well, how many healthcare workers are in your studies? How many who are in frontline retail? They've got almost 2,000 participants. So I think this is really unprecedented, but it is critical as we're communicating vaccines that this transparency is out there, that this is the kind of stuff that as the manufacturers share this, and some of them are a little better about sharing it this way than others, we will increase our confidence. And so, of course, the big question is, well, what about kids? How come we're not hearing about vaccines for kids? We probably won't see a vaccine for children until well into 2021. There are some that have enrolled children, but they haven't really progressed. So you can see the list here. Some of these are in other countries enrolling, just starting with teenagers for the most part. So let's just talk a minute about the different types of platforms. This is where we're going to get a little bit cellular for a minute before we close out. So there's these different types of platforms or ways that we can think about delivering and creating a vaccine against SARS-CoV-2. This is more the top upper left box is more the traditional way that you think about vaccines. Inactivated, where or live attenuated, where you take a killed part of the virus or a weakened part of the virus, and it basically mimics your immune system. This is an older technology. The main reason you're not seeing too many, really, I don't know if any American companies are pursuing this, is that it takes a long time. This is a slower, much slower process. There are some other countries that are using this type. And then down below, the protein-based, where we're used to that, the protein subunits such as flu, pertussis, Hep B, and also you can have a viral-like particle where you'll take a piece of that S protein, for example. HPV is developed that way. There's also what's called the viral vector, and where you've got either replicating, where you have like a weakened type of measles, where basically you're trying to get the virus to ride along with something that is friendly to the body and that will present the SARS-CoV-2 virus to the immune system. The Ebola virus is also developed this way. The blue part are the new things. So one of the new viral vectors is non-replicating adenovirus. It's new and has not been licensed. And then also these DNA and RNA viruses we've been hearing about for a long, long time. Was attempted for Zika. We've thought about it for HIV. We've thought about it for against cancers. So the concept is not new, but it is it's a rapid way to make vaccines, which I think is why many people have their hopes on this. This is the Moderna platform. It's good because it's fast. It stimulates T cells and B cells. It does require more than one dose, and the extreme cold makes its distribution kind of challenging. So this is, believe it or not, in looking at all these different websites to explain this to families or yourself or your colleagues, the Washington Post has some beautiful cartoons of the immune system and how these, what I just talked through, how they work. So you can see here the weakened or killed goes in to the cell, replicates, goes to the antigen presenting cell, and then over to the T and B cell. Whereas the viral vector, you can have this, the replicating viral vector or non-replicating, they'll go into the cell, and then the vector, whatever type of vector is used, and in some of them are like insects, a baculovirus it's inactivated, it is harmless, it's a fairy gene, if you will, that it carries it into the cell and then dissipates. The antigen then is again presented through that viral peptide to T cells and B cells. And this is the AstraZeneca product, is in phase 3 for this type of viral vector. And then again, you can see DNA, which is that outer protein, and the RNA, that inner genetic material, getting into that spike protein here, and then again to the antigen presenting cell. This is just such fascinating technology, and I am happy to say that a lot of these leading vaccine scientists are women, and really using each other well in how they're trying to extract virus, come up with new techniques, and really collaborating quite amazingly across the world. And then this is that subunit protein particle where you'll take either SARS-CoV protein, these sub pieces, little pieces of that S protein here, or you can take an empty shell virus that looks like the virus, it's not harmful, and you basically kind of trick the immune system a little bit into something that it recognizes. This is the Novavax product that is making the most progress. And then we have, after you've done the research, you have to decide who is going to get this vaccine. It will be a tiered approach. We will not have enough as soon as it comes out. It'll be about, there's 12 million healthcare workers and other folks that will be in that first tier, and then on to other essential workers up until we get to the general population. So you can see It's not going to be an overnight, the whole country gets it, and that's where we'll have to be good communicators about being patient. We prioritize the populations through modeling, which will get us the greatest gain and the earliest. We have to keep our healthcare professionals healthy. We also have to use healthcare professionals that can advocate for, like, I got the vaccine, I did really well with it, I think you should get it. And so it's in a really important population that we'll start with. And then the allocation process going out to the states, the distribution from the manufacturer to distributing sites to states, and then where will it be administered? At first, it'll be probably a pretty controlled, closed pod models in most states, and then it'll open up as time goes on to more broad use. And this says basically the same thing. I'm going to just show you this one because it's easier to read. Phase 1, again, we think of these as the jumpstart phase. These will be our high-risk healthcare workers, so people who take care of patients with COVID, first responders, but also we're really thinking more about will we have reduced infection and have a better outcome if we focus on our older adults as well. And so there is some discussion about moving them up to that phase 1A. And then phase 2 are other frontline folks, critical workers, teachers, people with of all ages with comorbid underlying conditions. Phase 3 then we'll get into younger adults, children, others, and then last will be pretty much everyone else who we haven't gotten. Throughout this whole aspect, though, will be equity. We want to make sure that we are not being inequitable, that we want to make sure that all communities get it, all countries get it. And so there's a lot of international discussion and collaboration going on. The FDA has done some scenario planning. You can see we're a little past this, end of October, and a little past, we'll probably won't make that end of November. Sorry, I think someone's
okay. I hear the rumbling, so I know it's time to go.
Oh,
Thank you for checking on me.
I think someone is not on mute. Okay, so the FDA has done quite a bit of modeling and looking into how we can roll this out. We're going to roll it out ethically. It'll be rolled out with these principles that ACIP has been talking about, which is maximize the benefits, minimize the harms, promote justice. We've rolled fairness into that, mitigate any health inequities, and promote transparency. So one of the questions is, how is this going so fast? And normally vaccines are sort of like a track that you do one hurdle at a time, go back to the regulator, go back to the manufacturer. Whereas this, there's numerous things that are happening at the same time, lots of collaboration, regulatory and manufacturing happening simultaneously. The safety is really the main emphasis here, that we have to have a safe vaccine. And I really like Dr. Grace Lee, who's on ACIP and on the COVID vaccine work group. She said, safety is not the absence of risk, it is the balance of risk. And so that's where we're taking our time to make sure that it's safe and effective. And then we're rolling this out for the people who are in the studies, and having them be able to communicate with their smartphone to the CDC. And when this first comes out to the community, we'll all be able to do this, to text message if we have any side effects, like a written VAERS report, but it will be able to be logged into the CDC by text. And then last, we're really emphasizing how we're communicating about this, communicating the vaccine confidence. And this is probably one of our biggest hurdles, because we have to have a high uptake, to have a vaccine that is really good and high efficacy in that 80% range would be wonderful. The lower it is, if it's in that 50% range, the higher number of people that we have to have. And we know how poor the United States is on getting our flu vaccine. So we've got our work cut out for us on this. The other thing is speed. If it takes us a long time to get everybody vaccinated and the season moves off winter and onto spring, this will likely be a winter virus. We may end up starting all over again. And so getting it out and getting that high community immunity as soon as possible is really our goal. And last, like all vaccines, phase 4, after it's rolled out, we continually monitor the safety through VAERS, the Vaccine Adverse Event Reporting System, through CISA, which is our Centers for Immunization Assessment, Safety Assessment, the Vaccine Safety Data Link, and the manufacturers do post licensing as well. So phase 4 studies are really, really important and will continue on. So if you want to be in a clinical trial, they still are recruiting. Here's clinicaltrials.gov, if you want to continue to monitor them, that's the website. If you want to be in a trial, that long one on the bottom, coronaviruspreventionnetwork.org, you can sign up. And then many of the slides that I've talked about in these upcoming ACIP meetings are available about 2 weeks after the ACIP meeting. And then this is just a little, we'll be talking more about COVID vaccines and COVID disease in a blog that we do at Children's, and I'm kind of a regular on it, talking pediatrics. So feel free to listen in. And I will end there and have some questions with my newly vaccinated 15-month-old who just got her MMR yesterday. Thank you, guys.
Thank you so much, Patsy. I know we're all giving a virtual applause to you this evening. So thank you. And we certainly have about 10 minutes or so for some question, and you had done such a wonderful job addressing some of the pre-submitted questions this evening. And I think maybe what we can do is I'll look to any questions specifically in the chat if anyone has any, but maybe what we can do is underscore some of the key points that you mentioned as it relates to pediatrics and also just the vaccine distribution and the process. So you had started to talk about for pediatrics, whether there'll be a recommended age of the vaccine, and where most of the trials right now are 18 and above. I think there was a comment from a colleague in the chat that the Pfizer study may now be involving children as young as 12. And so at this point, it sounds like we don't particularly know what the recommended age may be and what that would look like, as we're just starting to really get that data. Is that correct?
That is correct. And our recommendations come from where our studies were. So if we don't study down to 12 or to 6 or whatever, we won't be able to make that recommendation. And so watching those age groups, and as Dana points out, these younger ages are going to be slowly added. As we do okay with adults, do okay with young adults, we'll add teenagers, and we'll go down and add to the younger ages. Kids are different than adults, though. Their immune systems are very responsive, so we don't want to have any kind of immune-mediated adverse reaction, or we don't even know, would it potentially develop into an MISC after the vaccine. So everyone is talking about children are an important population, but we are going to go slow, and safety will be the focus.
Okay. And I know, Patsy, you've done so much work in vaccine hesitancy and working with patients and families that are that way. And since we don't particularly have a history yet, of course, of the evidence of this vaccine compared to so many others that have been studied, what are your suggestions as we get there in terms of just working with our patients and families that may be hesitant, knowing we don't necessarily have that history behind us?
Yeah, I would totally acknowledge with families what you just said, is that, this is, we're all on uncharted territory and we're on it together. And we don't know what these vaccines, how they will be, but we do know a lot. We know that research is going to give us the answer. We do know a lot about how viruses work. We know a lot about how we measure immune response. We know a lot about how we can compare a COVID virus vaccine to other viral vaccines. So we will have a lot of principles that we're going to lean on. And but the main thing is that the scientific process is strict, and we approach that with scientists are inherently skeptical. And so we will allow that skepticism to have us question and have safety as our guide all along the way.
I like that perspective. That's great. And you had mentioned and mapped out so nicely the sort of different types of vaccine platforms, and perhaps this innovation around the nucleic acid vaccine platform that Moderna is doing, and moving forward with that kind of speed that it's giving us. So we are getting some questions again of what's your sense of the safety as we explore these new platforms for vaccine development? And it seems like we're moving very thoroughly through the process, but if you could speak to that.
Yeah, I mean, we're all looking at that data very closely because it is uncharted waters. We have not had RNA vaccines before, and so watching how those studies go, having enough numbers, making sure that those are significant. So even in that antibody layer that I showed you from the Novavax paper, there were only 25 participants in each of those arms. So those are the kinds of things you look at when you're reading a paper: is what's the N, and how was the outcome, and getting it to a population. Was it a diverse enough population? Were there enough people that had underlying health conditions, et cetera? So, you know, we're all cautious, and I know some people have joked and said, well, I just need to see Paul Offit get it, and when he gets it, I'll get it. So he and I were just texting today about the election, but he is on the VRBPAC, and I just think everyone really respects his knowledge. He's developed vaccines. He knows the kinds of questions to ask. So the right people, I think, are at the table to watch those kinds of safety concerns.
Okay, and I see some questions looking a little bit also at age range. So we know we don't quite know what that'll look like. We also know that if children are really going to be in that phase 3, time frame, that we do have some time to sort of see what types of reactions or outcomes will be there, and that may be to our benefit in helping determine the age range. And so if you could speak to that, and also just in terms of children that are with chronic disease or those that have oncologic processes or things like that, would they be stratified any differently as part of this?
Yeah, I do think that, I mean, children realistically, I don't think we'll be getting the vaccine in the general population until late 2021. I think we do need to march through these other populations, study them, look at how they're doing. In those populations in adults, they are looking at folks with comorbidities. Right now, the early enrollees, there were not as many immunodeficient, immunosuppressed, pregnant, so those are things that we are going to need to add, but you don't want to start with those populations. You want to add them in after you see that the general population who are immunocompetent can handle that. So it's immunocompetent, young adults, middle-aged adults, and then getting into immunodeficient or adults, and then into the pediatric population. So it's pretty stepwise, and that's why kids are not going to be the first up. Also because of that epi that I showed you, that children have a much lower incidence of this disease and a much lower incidence of death. And so focusing on where our highest morbidity and mortality is another reason why.
There's a question from our former president, Dr. Landecki, who we all know well, looking at who said she was part of the polio epidemic. And so do you predict a similar sort of abrupt end to COVID as it was with the vaccines then, or will this look a little bit different?
Yeah, Linda Landecki, my Minnesota neighbor and very good friend. Linda, it's a great question. So where are they similar? Their viruses, their world, global, disasters. They're really spread differently, and I think what's making this just so difficult is this respiratory transmission. And not only that, so our first SARS-CoV-1, there was not pre-symptomatic or asymptomatic spread. You only spread the first SARS once you were sick. So you could identify people and isolate them. That is the hardest part about this. So it's hard to answer your question, Linda, because knowing who even has this disease is what's so difficult. So there's some neat technology coming out, like breathalyzers. So you can say, oh, I don't feel bad, but oh, I tested positive on my breathalyzer, so I got to stay home from work today, trying to identify people early on. I don't think there's going to be an abrupt end to this, knowing how coronaviruses act. You know, we've got the bad SARS, MERS, and SARS-CoV-2, but we've got the 5 other, you know, regular coronaviruses. They develop antibody protection for maybe a couple years, goes away, comes back again. And so this very well may be like influenza, that we have to deal with it every winter, and keeping a vaccine along with influenza. So we don't know that yet though, but that's a possibility.
Thank you. There's a lot of great,
sorry.
no, there's a lot of wonderful questions about equity. And I think you spoke so well to how these trials are trying to be designed, and the committee is really looking at ensuring that that is in place for so many of our vulnerable populations that we know, and we're trying to mitigate those inequities. Given that you said some of these vaccines are going to require freezing and those temperatures for their viability, how might that work with respect to equity of the availability of vaccine if those conditions are required?
Yeah, my understanding, and I think there's some folks who work at the state level who are welcome to chime in, but my understanding is that they are going to be delivered in these kind of super special freezers to the state, and then the state will break them up into sort of smaller, more manageable freezers. We've been asked at our hospital, can you take a smaller freezer on your dock, and is your dock at your hospital, your loading dock, is it secure? And so the worry about, you know, somebody stealing the vaccines or getting into them in some way, but it's this large freezer container that would be kept on your loading dock. Then you would take smaller amounts of that in with dried ice. And there's some rules around it, and it varies by vaccine. Once you take it out of that, how many hours you have to use it. So there's a lot of detail on each one of these vaccines, and in the storage and handling. And I know states right now are just working madly at some of these this micro planning, and how, because once we get approval, everyone wants it to go, like get it out there, get it going. And so the recipients in the states are really working hard on this.
Know. And I know we're almost out of time. I just want you to know, Patsy, what wonderful comments you've received from so many of our attendees this evening. This was extraordinarily informative and an excellent presentation. And
thank you. I'll have to read them later. I'm just looking right at you. I'm trying to pay attention to you.
Oh, no. Well, just a great presentation, just wonderful, so informative. And if I may ask you, Patsy, just one final maybe remark. I always like to ask somebody like you, who's a PNP and a leader in this world, what is one of the greatest things you've learned from the experience so far of following COVID-19 and our experience of what this is going to look like? What's kind of one of the lessons you've learned from the process?
Well, I've learned a couple of things. I've learned a lot. So I was the deputy incident commander. Now I'm the incident commander. I got a title, but not a raise. More work. So I'm in charge of all of the hospitals. Our 2 hospitals in Minnesota. I've learned that it is such a team sport, that you really have to rely on others. You have to collaborate, you have to listen, and it's so many skills that nurse practitioners have. We're good listeners, we're good leaders, we understand the science, but we can speak English. We know that it's, you know, to be respectful of all the different people. Like it has been such a team sport getting through COVID. And so I really think that the other thing I've learned is just being at ACIP and knowing that we're one of the last stops, and there's 15 voting members and 45 liaisons. NAPNAP has a seat. We are one of those 45. And I think our organization, with their foresight many years ago, submitting my name, and not just me, but just saying we have to have more nurses at the table. We have to have more voting members. We have to have, and our liaison seat is secure forever. And so that role that we have as nurse practitioners on the national stage, because of so many people on this call, Dana and others doing such, Mary Beth and others doing such great work, It's been like, sometimes I go, how did I get here? What am I doing here? You know, and then you think, well, no, I'm following this. I get this. You know, we have things to bring to the table. So to have that confidence and knowing that our experience, our education, our background as nurse practitioners in that practical sense of vaccine delivery, communicating with parents, understanding the science, being able to really analyze a literature, a research article, you know, we deserve to be there.
Oh, thank you so much, Patsy. I mean, I think it goes without saying you're not only just an outstanding leader in this field, but we're so proud that you are a NAPNAP member and that you are representing us at these critical tables, and you so much believe in the power that we have in nursing and in pediatrics, and such a wonderful pioneer and leader for us. Thank you so very much for sharing all of this incredible knowledge. I think we're all waiting with bated breath to see what this next year will bring, but this really gives us a great place to start from and learn going forward. So thank you again. Thank you to all of you for being here this evening, and we will make sure that we get this presentation together from Patsy with the recording for those of you who would like to listen in again. And if there are questions about tonight's town hall, anything, please email marketing@napnap.org. We put together a whole site on our website of resources on coronavirus in general. So if you visit napnap.org/coronavirus-safety, that's where we have everything. And I know some of you may not be members yet of the association, so please visit the website. If you are a student, if you're a pediatric APRN, if you are a pediatric advocate, we would love to welcome you, and you can learn a lot more about being a member. So thank you again for joining us, Patsy. Thank you so very much from our whole community. We wish everybody good health, well-being as we head into the holidays, and hope to see you again very soon. Thank you so much.
Thank you. Stay safe, stay strong, everybody.
Good night, everyone. Thank you.
Good night.